Your brain is the most protected vault in the known universe. It is wrapped in bone, cushioned by fluid, and guarded by a chemical wall so tight that roughly 98% of potential medicines cannot even get through. For decades, we thought we knew every guard at the gate. We focused entirely on the blood-brain barrier, assuming it was the only thing standing between your gray matter and the rest of your body. But in 2026, researchers discovered something we had completely missed. Tucked away in the choroid plexus was a second, independent defense system, the base barrier cells. Think of the choroid plexus as the brain's internal plumbing factory. It is responsible for producing the cerebral spinal fluid that bathes your entire central nervous system, keeping it buoyant and clean. Until now, we thought this factory was relatively open to the blood supply, but the base barrier cells act like a secondary security checkpoint, filtering exactly what gets into that precious fluid. It is a brilliant piece of biological engineering that ensures the brain's environment remains perfectly stable, even when the rest of your body is in chaos. The discovery of these cells changed everything we knew about brain anatomy. It turns out the brain doesn't just have one wall, it has a multi-stage airlock system. This isn't just a passive filter, it is an active mechanical system that uses specialized proteins to lock cells together with incredible precision. And the most fascinating part? We found that these cells have a unique mechanical signature that allows them to sense pressure changes in the spinal fluid. To understand how this works, imagine a high-tech airlock on a space station. You cannot just open the door. You have to equalize the pressure and verify the contents of the chamber first. The base barrier cells perform this exact function for your brain's fluid. They ensure that nothing enters the spinal fluid until it has been cleared by the cell's internal sensors. This explains why so many drugs that pass the blood-brain barrier still fail to reach their targets. They are getting stuck in the second airlock. But here is the clever part. By identifying these specific cells, we have finally found the smart gate of the brain. And if we can find the key, we can unlock it. This discovery is opening up a whole new field of barrier engineering, where we look at the brain not as a sealed box, but as a series of managed ports. Next, we are going to look at the actual molecular rivets that hold this system together and what happens when they start to fail. The secret to the base barrier cell's strength lies in its tight junctions. These are essentially molecular rivets that cinch the membranes of neighboring cells together so tightly that not even a single ion can leak through. Imagine two sheets of rubber bolted together every millimeter. That is the level of mechanical integrity we are talking about. It creates a waterproof, airtight seal that is virtually impenetrable. These rivets are made of specialized proteins like claudins and occludins, which weave through the cell membranes like a needle and thread, stitching the barrier shut. But the base barrier cell isn't just a wall, it is a smart gate. It uses a process called transcytosis to selectively grab specific molecules from the blood and pull them through to the other side. Think of it like a high-security revolving door. It only lets in people with the right ID badge, and it only moves one person at a time to maintain the seal. This mechanical precision is what allows the brain to stay nourished while keeping out toxins that are only a few nanometers in size. This system creates a massive chemical gradient. On one side, you have the blood, filled with proteins, waste, and fluctuating hormones. On the other, you have the crystal-clear cerebrospinal fluid. The pressure difference between these two fluids is managed by the BBC's mechanical sensors. If the pressure in the brain gets too high, these cells can actually adjust their permeability to help vent the system. It is a self-regulating engineering marvel. The brain literally manages its own internal hydraulics through these microscopic smart gates. However, even the best engineering can fail. In certain conditions, the tight junctions can leaken, leading to what researchers call the leaky valve effect. When this happens, the brain's internal environment becomes contaminated. This leakiness is now being linked to everything from chronic migraines to early-stage neurodegeneration. Understanding the mechanics of this failure is the first step toward fixing it. If we can reinforce these molecular rivets, we might be able to plug the leak before damage occurs. So how does this differ from the famous blood-brain barrier? While the BBB protects the blood vessels themselves, the base barrier cells protect the fluid-filled chambers of the brain. Think of the BBB as the security at the front door of a building and the BBC as the airlock leading into the server room. Both are vital, but they serve very different engineering purposes. By understanding this two-tier defense, we are finally seeing the full blueprint of brain security, and that blueprint is the key to the next generation of medicine. For a century, the delivery problem has been the graveyard of brain research. We have developed incredible drugs for Alzheimer's and Parkinson's, but we simply couldn't get them into the brain in high enough doses. We were trying to force our way through the blood-brain barrier, which is like trying to break through a reinforced concrete wall. It is designed to stay shut. But the base barrier cells offer a different strategy. Instead of breaking through a wall, we can try to walk through a door. By targeting the specific receptors on base barrier cells, we can trick the SmartGate into letting our medicines through. We're essentially giving the drug its own security clearance. This would allow us to use the brain's own fluid production system as a delivery highway, carrying medicine to every corner of the central nervous system. It is a revolution in pharmacology. We are moving from brute force delivery to precision engineering delivery. This could change the timeline for treating neurodegenerative disorders. If we can deliver clearing agents directly to the fluid where toxic proteins build up, we might be able to wash the brain clean. Diseases like Alzheimer's, which are characterized by the buildup of plaques, could be managed by simply keeping the brain's airlock and plumbing in top condition. We are no longer just treating symptoms. We are maintaining the mechanical infrastructure of the mind. Looking even further ahead, we might one day develop synthetic barriers or biohybrid cells that can be implanted to replace damaged ones. Imagine a world where a leaky brain barrier can be patched with a simple injection of smart rivet proteins that automatically find and fix the gaps. This isn't science fiction anymore. With the discovery of the base barrier cells, we have the map. Now we just need to build the tools. The next time you think about how your brain works, remember that it is not just a collection of neurons. It is a masterpiece of fluid dynamics and mechanical engineering. From the molecular rivets of the tight junctions to the smart gates of the base barrier cells, your mind is protected by a system more advanced than any airlock ever built by man. And now that we finally know it's there, we can begin to work with it rather than against it. The future of the human brain is wide open.